we're going to have three very interesting talks on how to look at cancer, different approaches. All these three happen to be portfolio companies of R42, looking at different ideas on how to actually approach three people talk about their particular ideas. Where Paul Finn AI. This is available online.
Sorry, I think Rajan's internet has been a little bit slow. Let me ping him. Let me try to ping him. Yes.
It's all right. Can you hear me now?
I can hear you now, finally.
Okay, great.
Yeah, I think the
So
last part over here is the R42 Institute project.
Slow today. Well, let's get straight to the there's a few conferences you can see us at. Stanford two, and there's more papers on aging. How to conquer cancer. And let's get straight to Paul Finn, and hopefully he's got a better internet connection. So
okay, thanks.
Let's hand over to Paul.
Yeah, thanks, Ron John. I hope you can hear me. And just bear with me while I share my screen. Okay, so could someone confirm that they can both hear me and see the title slide?
Yes, both.
Yes.
Okay, super. Well, thanks very much, Ron John, for the invitation to speak about this this topic. So what I'll be talking about is generative AI methods for use in cancer drug development across both biology and chemistry. I'll start with some of the biology activities that are ongoing currently, then turn to the area which is of most interest and relevance to my own company, Oxford Drug Design, which is developing new drugs for cancer, highlight some of the issues and the opportunities there, and then finish off with some general conclusions. So as I just very simply did a PubMed search for publications that contain the terms both generative AI and cancer, and as you can see in the graphic, there's been there is a rapid growth in the last couple of years in that topic. This search was very simple, and I'm sure misses a lot of the activity that's going on, but I think it is it does give a fair representation of rapidly increasing interest in this area. And when we look at the contents of these publications, then I'd say that most of the applications are in the area of either biology or the development of drugs in a clinical setting, which seems to be a very active area at the moment. So just some examples. These are examples just taken from recent releases into the public domain, either press releases or publications. They're not designed to be representative, but they give you a feeling for the sorts of things that are going on. So GE Healthcare, obviously a big company, recently announced a new product, this called ConcR Intellect. This is an AI system that collates all the many sorts of documents that are around in healthcare. Obviously, many of those are text documents and patient records and so on, but also images that come from analytical tests, et cetera, and assemble those in and present them to the clinician in a manner that helps support assessment of those patients' treatments. Can give some guidance as to potential for those patients to be enrolled in clinical trials, can alert if there are divergences from treatment regimens and so on. Initially, this is quite narrowly focused, I guess, on prostate and breast cancer, but GE Healthcare have said that will rapidly be expanded to other areas. So this is really looking to do something that can actually be real world help with oncologists treating patients. Some other applications that are in development vary greatly in scale. So at that one end, there's BioNTech collaborating with InstaDeep. They have decided that the best thing to do is to build an in-house supercomputer cluster to run the generative AI methodologies on. I'm not going to go through the details of the specification, but it's an incredibly powerful machine, significantly powerful, compared to others that are available worldwide. Of course, these very, very large generative AI models do require enormous amounts of computing time and extremely large storage requirements. So they've decided to invest in getting one of those themselves rather than using cloud services. So they're very active there and putting a lot of money in, clearly, and clearly also investing in developing the technology. So they're active in developing new generative AI methods. This Bayesian flow networks method is something that they've talked about recently, which they claim has advantages over the existing methods. And from what you can tell from the public domain, they're mostly using this in the area of bioinformatics, structural biology, sequence analysis, and so on. Obviously, BioNTech are very, very interested in developing mRNA vaccines for cancer, something that predated their interest in mRNA vaccines for COVID, and which they're still very much involved in. On a much smaller scale, again, this is just an arbitrarily chosen, smaller company, Triomics, that they have published some stuff on the OncoLLM. It's a cancer-focused large language model that they're developing to help, again, in this area of patient record interpretation and clinical support. And they claim that they can by developing a domain-focused model, they're able to get as good or better performance as the very large general purpose models, but at much reduced scale and cost. And finally, just at the level of sort of research ideas at the moment, there's a recent paper, again, you can see the references at the bottom, a proposal for really what is a framework, which they call OncoGPT, to predict the trajectory of the disease and help make decisions. So as we know, cancer is a disease that changes over time, that the tumor evolves and develops, and certainly under treatment and selective pressure, it mutates and becomes resistant and so on. So this is something which changes longitudinally over time, and the idea is that this framework could be developed to help identify those factors and alert the clinician when perhaps a change in treatment regime might be warranted because of the changes in the response and other things from the patient. At the moment, this is just a theoretical framework. A lot of work would be needed to be done to convert that into a real-world system that addresses clinical validation of this system, issues of data quality and quantity. Explainability is an incredibly important aspect of the application of AI in healthcare, where clinicians and the patients need confidence and trust in that the system will make sensible decisions, and having an understanding or an explanation of why the AI is making that recommendation is obviously helpful in that regard. And of course, as always, there are the ethical concerns to be considered. So a lot of work in the biology and clinic. There's also a lot of work ongoing in the area which is more relevant to our work in Oxford Drug Design, which is in developing new compounds via these methods that could be developed into treatments. And that's also an area that's rapidly growing in many aspects of the chemistry applied to cancer and indeed to wider therapeutic areas. And to give a snapshot of that activity, there was a recent interesting publication from Boston Consulting Group. So what they did is identify what they call AI native biotechs, by which I think they mean biotechs that have demonstrated or at least asserted that they use AI methods as a key component of their discovery activities, and then they decided that all molecules or all projects in their pipeline were going to deliver AI discovered drugs, and then from that set they analyzed that clinical pipeline in various ways. And I show two graphs taken from the paper on the right hand side. So in the top figure you just see the number of AI discovered molecules in clinical trials by this definition, and you can see that is growing growing quite rapidly, with, as you might expect, most compounds in the earlier phases of clinical development, but as these projects mature, some growth in the later phases as well. And interestingly, also at the bottom graph, that same data, but analyzed via the what was the key contribution of the AI to those projects. And you see that the largest one numerically was the AI contributed to target discovery, but which again I class in the biology side of drug discovery work. But there were a reasonable number where the AI had been involved in the design of the small molecules that had entered into the clinic, and then to a lesser extent also activity in antibodies, vaccines, and repurposing of other molecules for one indication into a new indication. It's another thing which they touched on in this analysis was, are the drugs that are developed by AI methods more likely to succeed in passing through the stages of clinical trial than drugs that are discovered by traditional means? And I think, unfortunately, the conclusion is that the data set is not yet big enough to be able to make a firm statistical answer to that question. So far, only 6 projects and 6 compounds, just looking at the small molecule drugs, have finished phase 1, of which 4 have progressed further down the pipeline and 2 have been unsuccessful. That is a higher success rate, I guess, than the industry average, but the sample size, I think, is too small to draw any conclusions. I think it's obviously difficult to understand and to get information for exactly the contribution of AI to each one of these projects, and I think one would that remains a very open question. But nonetheless, despite those limitations, I think it's giving some interesting insights into what is going on in AI in drug discovery. Of course, these are across all projects, but a great number of these are indeed cancer projects. So there's clearly a lot of a good reason to be interested in using AI in this area. If we think about the current technologies for large-scale on-demand chemical synthesis, which have expanded enormously in the last few years, there are now libraries containing billions of compounds that are available for synthesis on demand. And of course, you don't want to miss out on finding one of those compounds or identifying one of those compounds if it could be useful for your project. However, the size of these libraries are such that you can't store, there's not enough computer storage on the planet to store individual representations of every one of those compounds, and there's certainly not enough computing power to search through them in a dumb, systematic way. So it's very appealing to think that AI can suck up all of that information, process it, and then identify those small number of compounds that would be very useful for your particular project. That of course is the ideal. To the extent to which that can happen is still to agree an open question, I think. And certainly if used in an unconstrained way, the generative AI methods may sometimes have what are called hallucinations, which are just mistakes or errors, and think sort of far outside the box and come up with molecules that are difficult to make or otherwise have undesirable properties. So that's something that needs very careful consideration when using these methods. And it's very hard to know what is going on inside companies, as so little of that information is published, but one can get some idea from looking at some of the work that has been published. So I think it's quite striking that when you look at some of the publications, these generative AI methods are able to construct libraries of maybe tens of thousands, in some cases hundreds of thousands of compounds of potential interest. But when you look at what was actually made out of those libraries from the publications, it's really very small numbers of compounds were actually synthesized and tested, and that's quite a discrepancy. And that could be simply resources. These often done in small companies who don't have much money to make compounds, but these numbers still seem quite small. So one wonders whether all of this effort that's going into the generative AI is largely wasted in making molecules that actually, for the project team, are not that interesting. And I think there's one example where that would appear to be the case, which is a nice example, which I'm going to throw on this slide. This is work published by In Silico Medicine. It's a few years old now, but I think it's still an interesting learning example. They had developed a generative AI method, which they call Gentle, and they applied this to their project of interest. It was a kinase cancer project, and they looked to get this method to make compounds that were synthetically feasible, novel, and had the biological activities that they wanted. And when you look into the details of this paper, the generative AI method generated 30,000 novel compounds, but those compounds were heavily triaged as the next step in the process. So over half of them had inappropriate properties and were lost at that point. Some of them had maybe undesirable other medchem features. They weren't sufficiently diverse. They were already available as commercial analogs. And then there were some product or target related filters that they applied, including synthetic accessibility, so that they only made 6 compounds out of that set. Now, on the positive side, two of those compounds were validated in cell-based assays and were actually quite potent. So you could say that the generative AI has done its job. On the other hand, this does seem to be enormously wasteful in terms of the effort going to build the models and the large amount of human triage that's necessary in order to get to that end. And furthermore, it was pointed out that for one of those 2 compounds, it bore a striking resemblance to an existing compound with activity against that target, which in this particular case was actually in the data set used to train the generative network. So just because the compounds are novel doesn't necessarily mean that they're going to be that interesting. And I think as a result of this, the methods that have been developed more recently have included features in the network model to move the generative network away from molecules that are already known in order to increase the synthetic diversity and novelty of them in the future. Another example that's also quite instructive as to the way in which these models can, if you're not very careful, lead to artifactual conclusions. This is some work that was done by workers at MIT a few years ago. This wasn't in cancer, this was in antibacterials, but I think the principle is still relevant. So they used FDA-approved drugs and some natural product library to train the network. They then predicted compounds from a different database to see whether they would be predicted to be highly active. And they tested 51 of those, and one compound that came up as being of interest was a compound which they called halicin, the structure of which shown on the slide. And digging into this paper is also quite attractive. So halicin, they determined, had a novel mechanism of action, not present in any of the compounds that were used in the training. There were no related chemotypes that were antibacterials in the data set, and yet it still managed to identify this molecule. And I, with my colleagues, were interested in trying to understand how the neural network could have identified this novel mechanism of action compound from the information that was given in the training set. So we looked at this data from the perspective more of structural fingerprints, chemotypes, to try to, because the neural network, of course, is a bit of a black box. We wanted to try to see if we could understand this using different machine learning methods where you could get some understanding of why the method was making the prediction. And what we noted was that, in the there was an imbalance in the training set. The actives in the training set contained sulfur atoms far more frequently than the inactives. So we were suspicious about that. We modified the data set by removing one class of potent antibiotics, the beta-lactams. All the beta-lactams contain at least one sulfur, so that if you remove that beta-lactam class of antibiotics, sulfur is now roughly equally represented in the actives and the inactives, and halicin no longer predicted to be active. So we strongly suspect that although halicin was predicted as an antibacterial, this had nothing to do with it uncovering a novel mechanism of action or anything of that sort. It was simply a bias in the data set that sulfur was more prevalent in the actives than in the inactives. And you could, possibly using modern techniques, you need to check for those biases, and perhaps could have used something like the SHAP feature contribution analysis to alert you to that problem. So in chemistry, there are many pitfalls of this sort. I don't want to give you the impression that I'm poo-pooing generative AI. I think it's a really exciting area, and there have been additionally very positive examples of its use. So here's a positive example, I think, where it was done very well, developing macrocyclic CDK2 inhibitors, again for cancer, again using a machine learning generative AI method, starting with some compounds that were known actives from Pfizer and from others. They used an iterative approach to convert these molecules into the macrocyclic structures that they wanted. Macrocyclic structures are often have good biological properties in terms of stability and so on. So they're quite attractive, and they really, I think, used the generative AI really very effectively to come up with these very novel macrocyclic structures, which had the potency they wanted, a degree of selectivity against an off-target, and activity in ovarian cancer cell line. So a very nice piece of work. And I think it was notable that this was really a collaborative work between the computer scientists, computational chemists, and medicinal chemists to get this moving forward effectively. So we believe that to use these methods effectively to generate the hits you want, you have to use them in some sort of a constrained way that prevents them from accessing inaccessible chemistry, and make sure that they're producing the desirable properties that you want. And so briefly, just to talk about the approach that we're taking in Oxford Drug Design to do this, to meet those ends. So I think key is the chemical representation that one gives to the AI method. Many of the methods that have been developed for applications in chemistry use an abstract representation of the compound and project into a latent space where the relationship between that representation and the desired properties that you want are very hard to discern, and can make it very difficult for the AI to find those. So we prefer to use representations such as shape and charge distribution that are physically and physiologically meaningful, are related to the mechanism of action. These have fewer parameters, and we believe reduce the risk of overfitting. The other thing we do is to constrain the search space so that it's not so that all of the molecules that are made are synthesizable from with known chemistry routes, with commercially available starting materials. So we would start with data for a particular molecule, fragment it into fragments that can be resynthesized into different combinations. As I say, use these physiologically and physically relevant shape descriptors to populate an initial data set. From that data set, select those molecules where we expect to see the greatest expected improvement in our desired target function. Either compute or ideally experimentally measure those compounds, update the Gaussian process, and repeat to really optimize the activity that we want. And once we've achieved that, so say the key benefit is that each of those molecules can be made and tested relatively cheaply from known routes and existing starting materials. So one has to be very cautious and skeptical about AI claims. There was a lot of work done in the COVID times with people trying to look for these. Many of those studies were rather poor. There was no experimental validation. There's limited benefit comes from that, but some people did it well. Really using experimental validation, thinking about their models, and came up with some really quite interesting compounds. So in conclusion, as I don't think there is much time left, many generative AI models have been developed for improving cancer treatment, right from drug discovery through to clinical application and for patients. Many of those applications in the biology and clinical setting are being developed. They're being tested now in hospitals, and I'm sure will be extended and deployed more widely to the benefit of clinicians and patients in the coming years. Thinking drug discovery, that's at an earlier stage. It's a much more challenging area, I think, for generative AI. It's very difficult to validate these models. Unlike in bioinformatics, there's very often a lack of data. The data can be very sparse. That can make it very difficult to generate models, especially given the complexity of the structure-activity relationships between the chemical structure and the biological properties, especially when this is a multi-objective optimization, when the molecule has to satisfy many, many different, often conflicting properties in order to be a potential drug. Nonetheless, progress is being made. Not only ourselves, but others are moving towards more relevant chemical representations in their generative AI models, smarter searching of the chemical space to make it much more efficient and effective, and having more realistic goals, because these models are always, I think, best used when integrated with existing methods. And then I think we will start to see much more effective models emerging, and then these really being applied, and so molecules entering the clinic that have really benefited from these AI methodologies in combination, of course, with a whole other existing inputs into that. So thank you for attention. I just mentioned a couple of acknowledgements to colleagues at Oxford who have been involved in the development of the methods, Anthony Chabanaris-Assad, and a colleague at the University of Buckingham, Rishi Jagdev, who was involved in the Halocin analysis work. And with that, I will stop, and happy to take my questions either now or in the discussion session at the end.
So I'll sort of paraphrase maybe got time for one or two questions, because we started a couple of minutes later for you, so you get your full time, Paul. Hopefully people can hear the internet's working for me.
Yeah, it's coming through much better now, Ron John.
Okay, excellent. So I'll combine the two questions, my question and Panika's questions. So generative adversarial networks, generative AI. There's lots of types of different kinds of AI systems, and you said there's too many, generates too many ideas. Well, what does work? Are there other types of AI systems that you think maybe more promising, or subsystems? And have you done anything at Oxford Drug? Do we actually have anything at Oxford Drug that has been spit out, that's manufacturable, that works? And how does it work? At what phase is it at? Maybe you can just combine those two ideas.
Yeah. So taking them in order. As I say, I think a lot for me, a lot, is related to this sort of representation. How do you present that data to the generative methods? So I think the methods that are based on graphs, so graph convolutional networks. So I mean those map very nicely to chemical structure. If you're doing this at the 2D level, but I think that the thing which will work best is I think maybe decoupling the chemistry from the AI. As I say, I think searching these spaces where the molecules are synthesizable, rather than have the AI add atoms together in a way which optimizes the function, but then ends up with something that is either, well, they're all chemically feasible, but are PhD projects in order to synthesize. And so it's a shame that all of that effort gets involved, gets eventually wasted. So I think these constrained systems are the way to go. In terms of what we have internally in the company, in terms of these generative models, we have, I showed you little snippets of this system, which we call internally Synth.AI. We have, we're doing benchmarking work on that, where we have identified molecules with activity through that methodology. The molecules which we have that are most advanced and are approaching preclinical development were developed with the aid of AI systems, but previous generations, I guess, because it just takes a little time to go through the optimization to get to that phase. Hopefully that answers your questions, Ronja.
So you've got something that you've done at Oxford Drug that's moving through a pipeline of some kind.
Yeah. So we have our lead cancer program. We have compounds with a novel mechanism of action, novel chemical matter, activity in vivo, in multiple different models for reducing tumors, and are very effective in those preclinical models. And I'm hoping that 18 months from now or whatever, we'll have a molecule ready to go into the clinic from that work.
And what kind of cancer is that? Would that be for?
Because it's a novel mechanism of action, there are multiple opportunities. So this is impinging on a pathway with multiple opportunities. Where we have the most data is in colon cancer and in breast cancer. But I'm sure that the applicability is wider than that. But where we've demonstrated activity is in those two cancer types.
Excellent. Thank you very much, Paul, for being exactly on time and leaving time for some questions. Let's move to Professor Biankin now. So, Andrew, are you here? I think he is here. Last time, yes, he is. And
yes, I'm here. I was expecting to be third, so I've got a little bit of setup to do. I have a video to start with, so just bear with me for a couple of seconds.
I'll introduce you while you're doing that. So Professor Biankin is Professor of Surgery at Glasgow University. Is also advisor to ConcR. I guess more than advisor, really, sort of, I think, one of the original co-founders as well. And he's going to, I guess, can we figure out treatments? No, will the treatment work? Because notoriously, when people get given treatments for cancer, their life extension is not that long statistically for most of those things. And maybe, Andrew, you've got some ideas for us. Off you go.
I certainly do, Ron John. Can everybody hear me okay?
Perfectly.
Okay. So I'm going to start things a little bit different. I'm going to start with a little bit of a setting the scene video. This is something I've been working on for many, many years, and it's realizing the promise of precision oncology. And we believe that this can be realized by predicting an individual's response to a treatment ahead of time. Currently, we do things on the average, and the actual incremental benefit for most people is very small, if anything at all. And so by predicting an individual's response, we move past the challenges we have in sustainability of healthcare, because most of what we do in cancer and in many disease types, most treatments don't work for a given patient, but we don't know whether it's working or not, and we certainly don't know if it's working ahead of time. So I'm going to start with a little bit of a video here, and I'm going to adjust the size, the sound, because it's a little bit tricky. So I'll make this work. So I think it's pretty clear that we can do better. We have good treatments, but we don't use them as well as we could. Many of the approvals, and we read lots about this, the incremental benefit, particularly on a population level, is quite small, and we're starting to struggle to maintain this. In the UK, a quality adjusted life year needs to be £30,000 or less. Most drugs don't satisfy that. We have the Inflation Reduction Act in the United States. What about low and middle income countries? Don't have enough drug, but if you could predict ahead of time which patients are going to benefit, you don't need 100% of drug. You might only need 30% of drug for a drug that's approved. So let's think back. Can you believe that not that long ago we used to bleed people and do other barbaric things to treat them? How crazy is that? Well, bleeding still works. We still bleed people to treat them, but we don't bleed everybody that's unwell. We bleed people that have polycythemia rubra and hemochromatosis, and it works well, but only in those people. And so what's going to happen? How are we going to be thought about in 50 years' time? Can you believe that not that long ago we gave toxic chemicals to people to treat diseases without even knowing if that would work or not for that particular individual? And I would suggest that for most cancer drugs, even in the better developed targeted cancer drugs, most people don't benefit substantially. But how do we pick those people? We currently pick those people on average, and we choose how we do things based on an average response. We know cancer is complex. The more molecular detail we have, we understand that. But beyond those single gene events where we target specifically, we haven't made a lot of progress in better selecting patients for treatment. And you can see the archetypal approach of targeting a molecular process people often forget is the estrogen receptor in breast cancer. This was developed in the 1970s. That's 50 years ago. And the reason it succeeded is because 70% of patients with breast cancer are ER positive. And I would suggest that tamoxifen and its derivatives have done more for cancer care than all other treatments combined up to this point in time. So the principle of matching the right treatment to the right patient works, except we don't know how to do that. And certainly we don't know how to do that for chemotherapy. I work a lot on pancreatic cancer and have over the years. We have more and more drug options. Unfortunately, they don't work very well. And if you don't accidentally get the right one first up, then the patient usually dies. So what I want to start with is the power and benefit of predicting an individual patient's response to a given treatment, rather than the current trial and error approach that we take. And so let's think about this in the future in the health system. Where do we want to get to? We can't tackle this now, but where do we want to get to in the way we develop drugs to get there? So I'm a doc, I'm an oncologist. I've got a patient coming in, I've got 4 or 5 choices of treatment. Which one do I choose? Well, okay, the textbook tells me, well, the first one, treatment A, has a 55% chance of response, so we'll use that one first. B has 50%, C is 42%, D is 32%, and this is quite good for any sort of cancer. Normally it's a lot less than that. But every individual and their cancer is different. If you had your first patient, Mrs. AS, you can see her first treatment isn't going to work. So you're going to give her a treatment for 3 months, look at the scan, scratch your head and go, maybe we'll give it another 3 months. And at 6 months you go, oh, no, it's not working. Maybe we should change it. And you go to treatment B, and maybe she'll survive to treatment C, where the chance is 85%. Mr. JW, he's in luck. The first treatment works best on him. Ms. PB, the second treatment. And Mr. FAB, well, none of the treatments are working very well, but there's a clinical trial that's predicted to respond. So if we could change the way we approach this, think of the efficiencies and think of the patient benefits, the avoidance of toxicity on those drugs that aren't going to work. And that's why we're running out of money, and that's why it's unsustainable. Can we do this better? We don't have to get it 100% better, but even if we're 20% better, we're already going to shift the needle. And the last one is me, Professor AVB, and look at this, nothing works for me, and I'm thinking, you're going to give me these drugs, you're going to spend $1 million US on treating me, let's make a deal. You give me $500,000 and I'm going to buy all the white burgundy I love and enjoy the last 6 months of my life. And you save $500,000 as well. So how do we get to this? So we can't do this in healthcare, but we can start doing it in drug development. How do we enable this way of thinking? And we're already starting to do this when it comes to single gene biomarkers, and we know the probability of success is much greater. Signatures we're moving on to as well, but it's more complex than that. When we don't know the target, how do we move forward? And these biomarkers may be different for many different patients. There's a lot of diversity, and we don't understand the details of the biology yet well enough at this point in time. And we know we're not winning. The cost of drug development is increasing and increasing. The benefits are getting smaller and smaller, and oncology is a major offender. So what we need to do is to decrease the uncertainty and the risk as we start to develop therapeutics. Then we need to reduce the cost of R&D as well in order to be able to deliver therapeutics for cancer and potentially all other types of diseases in a way that's going to be affordable and sustainable in the future and more efficient over time. And the way we're approaching this is through creating digital twins of individual patients and their cancer. We're using Bayesian modeling to integrate complex data parcels and then try to move this into a way that enhances drug development. And we use disparate data points from a large number of samples. We do this so the algorithm learns, but when we actually do the analysis, we only need small data to be able to do that. This is a combination of data parcels that are publicly available, proprietary, through partners, through some of our clients and customers, etc. So that allows us to piece together units of biology associated with response and resistance. And many of these units of biology are through currently available clinical grade tests, like a large genomic panel or an exome plus a transcriptome, and the price point's about $5,000 US. So it is absolutely possible in this current time. We didn't create something new. We took astrophysics technology of decades of development that used algorithms that are based in Bayesian statistics to tackle a similar but unrelated problem where you're looking at uncertainty. You're looking for dark matter. You can't see it, but you can measure its effects. Similarly, in biology, we can't see what's actually going on, but we can do something to measure its effect. And we can learn from these data parcels and start to predict an individual response. We can do this preclinically and clinically, and I'm not going to spend a lot of time on the preclinical aspects of this, but what's most exciting for me, being a clinician and a translational researcher and a drug developer, is how can we do this in early phase trials? How can we get a better line of sight? How can we de-risk our go, no-go decisions and become more efficient and effective in the way we develop drugs and get them to market quicker and have a longer IP life? So the first question we want to know, does it work? And so let's look at this in the first context. So this is a data parcel, in fact it's TCGA. We ask the question, if we predict a particular response for a digital twin of a patient in this data set and predicted their optimal response, what would that look like? So if the patient received the treatment that we recommended, their response was 75%. But if patients didn't receive the treatment that we thought was optimal, they received another treatment, still approved, and reimbursed, their response rate was 55%, over 20% less. If you looked at what impact that has on survival, the purple line is the optimal prediction, and you can see the median survival benefit is over 2.5 to 3 years over this period of time, and the long-term survival is substantially better. So that's the first bit of evidence. Sure, it's retrospective data, but it gives us confidence that what we're doing is working. We then wanted to see if we could predict the results of clinical trials. We did this on 9 trials because we had the data to address these particular questions. We didn't select these 9 trials for any other reason apart from we had the data to be able to address this. The first one I'll take you through in a little bit of detail because I think it's quite interesting. So this is a clinical trial, very dear to my heart because I was the surgical and translational PI on this study. Margaret Tempero and I designed the study many, many years ago. It took 7 years or more to run, actually longer than that if you can include the designing process. 866 patients. I think it cost about $250 or $300 million to run this trial. And then we thought, could we predict the results of this trial? So the first step was that we took a completely different group of patients. I think there was about 180 odd patients. And we predicted the response of gemcitabine which was the standard of care at that time, versus Abraxane or nab-paclitaxel plus gemcitabine, which is what the trial was trying to do. If you see in the first curve, the orange predictions are the combination of the new treatment. The purple predictions are the original gemcitabine, and the first thing you'll notice, there's not many purple bits there. That's because they're hidden under the orange bits. But if you look at the curve on the right, which is the log odds ratio of the probability of response, you can see that only 30%, 32% of patients benefited, and a subgroup of those had a real benefit. And that drove the positivity of the study. So if you looked at this predicted curve here, you can see the top one is gemcitabine plus Abraxane. You can see the bottom one is gemcitabine. So 32%, that's a third of patients, are driving all of this difference. And look at the actual trial results. They look pretty close. So if you look at it in a different way, 68% of patients derive no benefit. Yet it's a positive study and this treatment is being used. So we can do better than that. How do we predict this group of patients' response? How can we design clinical trials? So the first benefit that we aim to do is to design better trials for success. The sort of parameters we do use at the moment is we estimate what the hazard ratio we want, then we do a power calculation, whereas if you have this predictive technology, then you can better design your trials. We can also start to define which of the 68% don't respond. We don't want to get it perfect, it will never get it perfect, but if you took away the 20 or 30% that are never going to respond, suddenly you've got a bigger signal, you've got greater patient benefit, and you've got a much better chance of approval. And this has obvious value all the way along the chain, including when it comes to healthcare. We also looked at another 8 clinical trials, and these are in metastatic disease, across different cancer types, across different treatments, both single agent and combination, and the tech team didn't know what the result was, and even if they did know, they wouldn't know what it meant. And of all the 8 trials that we looked at, we were able to accurately predict positive and negative results. So we're quite confident that it is actually working, at least in this context. And so where is this application? Where is it going to be most useful in drug development? By the time you're in a phase 3 study, you've already sunk all the investment into it. You've made a decision for whatever reason. It's challenging to stop a phase 3 study. What you want to do is decide which assets you're going to take further forward, which ones you're going to invest in, what's your clear go, no go, and then have a line of sight on what your pivotal trial looks like. And so there's a number of things we do, but this is the one that's of greatest interest, I think. And that's in early studies. So one of the challenges we have in early studies is that we have to do them in nth line therapy. Multiple cancer types, they've had multiple pretreatments. It's often difficult to get a signal. The cancer's a mess molecularly. You can't work it out. You want to try and do that a lot earlier in treatment, and that usually means combination with standard of care. And we used to do this a lot 20 odd years ago, and we could always struggle with deciphering what the contribution of the standard of care was and what the contribution of our new drug was. And we always went back to the average and said, let's look at the average. So this is a single arm trial, 30 patients. They've received the standard of care plus the new drug. And you look at it and you say, well, the median survival is 20.7 months, and the standard of care on average is about 20 months. And you think it doesn't look that different. But when you look at individual patient prediction, it is quite different. So the bars are the predicted responses of each patient to the standard of care backbone. The star is the actual response, and whilst you can see many of these stars fall on top of the prediction, there's a number of them the survival is much better. If you map these out on average, again, you've got these two survival curves. You've got your drug, which is the one on the right, the standard of care. You think, well, there's a bit of separation there, but the median survivals aren't that different. Do we take this drug further forward or do we not? But then if you look at individual patient prediction and you pull out those 10 patients, it's a third of these patients that did respond, you see a dramatic difference. And if you see this dramatic difference, then you can say, well, okay, these are the patients we want to take further forward. What is it about these patients? And you can dig into the biology of each one of these, and you compare those that responded versus those that didn't respond. You understand that, and you start enriching for responders, which increases your probability of success dramatically and gives you a much better line of sight on target for filing. So we know that the costs of genomic tests have gone down. For clinically available tests that we use to make these predictions, it's a reasonable price point of about $5,000 US, less if you get a discount. There's a lot of effort going in policy here. We've got Project Frontrunner. The idea being is that you want to start testing novel therapies in earlier lines of therapy. We're familiar with the Inflation Reduction Act because we just can't afford this current trajectory anymore. How do we change that? And the answer to that is prediction. We work with a number of clients, customers, partners, etc., to accumulate data and also help them move things forward. We have a number of applications of this far-sight twin technology, whether it be through synthetic controls to compare to their existing clinical trial data. And we do it in a number of indications where we start to be able to unpick some of the biology as well as define those patients that are more likely to benefit. We're also running our own clinical trial. This is called Vision, or AI Vision, which is in breast cancer. And in breast cancer, there's a number of choices of treatment, particularly for the deadly triple-negative breast cancer patients. Which one do you choose first? Because they may be dead by the time you get to the right treatment. So what does this mean? We can help with the best design for success through the approaches I've just suggested. And because it accelerates how we move forward, it generates value. So our overall aim is to change the current trial and error approach to treatment. We're tackling that through drug development for sensible reasons. And we envisage a day when each individual patient is given the optimal treatment for their disease. We think this should occur through better patient selection. We use various surrogates, we use clinical markers, etc. We need to move to better biomarkers. The current biomarker strategies have mostly been exhausted. We simplify things in development and hope for the best. We believe that we need AI at the bedside in the future to be able to better select an individual's treatment ahead of time. Initially novel drugs, and in the future for drugs that are already in use. And at that, I'll finish.
Excellent, Andrew. And that's Dr. Irina as well, your CEO.
Yeah, she's online. She's our CEO. Contact her. She's better at responding than I am.
I'm sure not. I will combine a couple of questions again, just to do one question. So I guess this is sort of the end user. Is really, do you view this as an individual, this AI at the bedside thing? Or do you view this as more of a drug development thing? It's sort of like sort of Josephine's questions. We seem to we may pursue some drugs, but we stop because of it's too expensive, or we stop because we've already spent 100 million on it and keep going. Sort of denial kind of approach. What's your view? Is this a bedside thing or is it a sort of industrial thing?
So it is an industrial thing at this point in time, but in the future, it's a bedside thing. We're not tackling the bedside thing. Healthcare is just too difficult. If you think of a super tanker, this, healthcare is like a planet We're not going to shift that. We're not going to change it for drugs that are currently being used. But if it's part of the way drugs are developed, we need to better select patients for the right drugs. We have plenty of great drugs. They work really well. Big pharma make really good drugs, but it only works in a small proportion of the population. We need to be able to get a better bang for our buck for each drug. So it is in drug development, but at some stage, it will follow those drugs that are being developed into the clinic.
Right. And then this idea that you're predicting, we saw several charts that predicted, you said the prediction was better than the treatment that was given. But how often is it the prediction is actually the treatment? It actually says just go ahead and use it. How often?
Yeah.
That wasn't quite clear that in the logic.
So we I mean, to test that, you need a prospective study where you're actually giving one set of docs the answer, the other set of docs not giving them the answer, and see what happens. That's a very long road. I mean, that's like testing anti-aging
Is
drugs.
there any intuition there, or?
So we have this data from that group of patients. We are working with partners for clinical trial data. So where they've done the tests and they have the results. So for us, the faster road is to not do a prospective study in existing drugs because there's not much value in that. For us is to take clinical trials that have been performed of high quality and then analyze those in a different way and say, okay, if you did it this way, this is exactly what would happen. Our road moving forward is through our relationships with pharma as they develop a drug. And then we don't have to pay for the clinical trial.
And then my final question is, because a big thing is that we don't need, in this approach, you don't need as much of data. I was in the UK a couple of weeks ago and I got a letter in the mail saying, would you like to be part of a 5 million person database collection and hand out your blood and your biomarkers? Do you see, this is an intuition thing, is it possible that if we collected all the data will solve our problem? Or is it really the body's so complex, it doesn't matter how much data you collect, you'll be, if you've got the entire population upload their genome and
Yeah.
Apple Watch data or whatever, and then medical, would that solve medicine, or really it's just still too complex? Intuition.
Well, it will if you collected every single possible variable on everybody that ever existed since Adam and Eve. There will be questions you could address. I mean, if you think of what happened with the COVID pandemic, we were looking for all sorts of drugs. If you had a database of people that were receiving common everyday drugs and who got bad COVID and who didn't, of course you can glean insights into that. But to be able to, I mean, if you're taking population level stuff and you're looking for a signal, and if it takes 5 million patients to detect a signal, I mean, what is that signal worth? Again,
Okay.
it breaks down to the individual. You'll find something, but some people smoke all their lives and never get cancer, and lots of people don't smoke and still get lung cancer. So it's down to the individual, this whole population-based approach. I mean, you can use Mendelian randomization and all these things.
Great. Thanks, Andrew. Thanks, Professor Biankin. That's great. Excellent. That was so interesting. That was such a clear presentation. I think that'll be useful for ConcR. Okay. Let's see. Let's go to Dr. Yasaman Kalantor-Motamedi with Agemica. And this is actually a company I co-founded with Yasaman. And really we set the task is, of a moonshot idea. A moonshot idea. Can you, and so Yasaman, while she's uploading her slides, can you set a concept of a vaccine for aging? A vaccine for aging. Now, what does that mean? So I'm going to hand it over to Yasaman, and she's going to talk about the cancer part of this project. Go ahead.
Hello, everyone. Today I want to present Agemica and our approach towards aging as well as cancer. I'm sharing my screen. Just a second. Yeah, can you see my screen?
Yes.
Yeah, so our mission is to increase the healthy lifespan of humans to 150 years via AI. And this is a company I co-founded with Professor Ranjan Nag, and we are actually creating a vaccine for aging. However, we are also breaking this aging vaccine down to finding a vaccine for chronic diseases that are the top killer diseases. First, we want to start by creating a vaccine for cancer. Then the biggest killer would be cardiovascular diseases. We want a vaccine to prevent that, and a vaccine for neurodegenerative diseases, and later on muscular degeneration. So the vision is that you take this vaccine in your 30s or 40s, and that would stop the aging process and or slow the aging process. And increase lifespan of us to 150 years by preventing us from getting any chronic disease. The problem we are addressing is that there is a lack of any preventative therapy or vaccine for chronic diseases. And that is not a small problem. You know, there's an old saying that says prevention is better than therapy. However, with the advent of medicine and the FDA approval process allows us to get drugs approved for therapy only, and the vaccines are mainly for viral diseases. Vaccines are emerging recently for just a small number of cancers, but there is no vaccine that prevents cancer and the rest of chronic diseases. And if you look at each of these chronic diseases like arthritis, heart disease, cancer, and diabetes, the incidence rate as well as mortality increases with age. So it's interesting to know if aging is an actual contribution to incidence of each of these diseases. And so if you look at cancer alone, in the UK we have 275,000 cases and globally 28 million incidents. Sadly, the cancer 10-year survival rate is as low as just 50%. And there is no preventative therapy. It is expected that 1 in 5, and some other stats says 1 in 2 of us get cancer sometime in lifetime. However, we cannot prevent that currently, and traditional biotech looks at each of these diseases one at a time, each subtype of cancer. However, we just miss the bigger picture of aging and the aging processes that are the root cause of getting cancer in the first place. So what is our solution? Agemica has an AI platform that enables us to bring data of all different diseases together and data of all drugs together, and give us predictions of therapies with high probability of success. So we developed in-house platform that is our private AI platform, and is our trade secret. We gave it data of 34 different cancer types and 175 different subtypes, and we gave it data of 1 million drug signatures, and it enabled us to analyze all the data, identify key genes and pathways that are disrupted in each of these. You can look at each of these circles as set of mechanisms identified for each of these cancers. But also we look at the bigger picture and identify what are the key mechanisms that are overlapping and shared across cancers. And those are the ones that if we could solve those mechanisms and pathways that go wrong, we could have therapies that would have the broadest possible effect across cancers. So those are the key mechanisms we want to address. And that enables us to identify a combination of safe drugs that are interesting for solving cancer and aging. And then we go ahead and do preclinical validations of the candidates with pan effect. And the targets that we identify are the key point for starting to develop and design a universal cancer vaccine, and that also enables us to create an IP for drugs as well as vaccines. But how do we know that it actually works? So we have first started by validating the platform in silico, and we gave it data of 34 different cancers. And in 17 of the cancers, just by giving data of genomics data, it could rediscover the standards of care for 17 cancers blinded. So it is important to know that the platform does not know what is the current care. It just looks at genomics data and spits out drugs. And then we look at those drugs and check and what is the standard. And in 17 of the cancers, we could actually rediscover current care blinded. The chances of that happening random statistically is 1 in 10 million or 100 million in some cases, which is significant. The next layer of validation is actual preclinical validation. We started by some in vitro validations in our first run, and we had 40 preclinically validated candidates in 11 cancers. And then we followed those predictions with CROs and in animal models, which I would present later. So our products start with combinations of drugs that are, we call it synergistic. That means that two drugs work together better than any individual drug. And that enables us to create IP on that. Our next layer of products is a universal cancer vaccine, and then later on we want to create an aging vaccine. So what happens is that our target customers are firstly cancer patients, and because two drugs are working so much good together, we would expect higher efficacy because the two drugs are creating a better effect than any individual drug in it. Plus, that means that we need lower doses of each of these drugs. That means we have lower side effects. And we are killing cancer in many different ways, and that means we are reducing chances of drug resistance. And the drugs are meant to be preventative and targeting those key mechanisms I mentioned. That means that we can prevent cancer metastasis, and that is a major problem in cancer. And then with our cancer vaccine, we can get it approved firstly for cancer patients for therapy, but then later on we want to expand its application to healthy individuals at risk, and one day we want to change the standards and make it a standard must-have vaccine for any individual at least 40 plus to increase health span of everyone. Yeah, that is a cool idea. We need to also make money out of that. So we have a business model that enables us to partner with pharma or sell our therapies. So if you look at each of the drugs that get approved, the sales go higher and higher in terms of billions, reaches like 12 billion, and then the patent expires and the sales drops sharply. And that is a common problem with drugs that get approved and pharma companies own them. And if you want to know how much big market size is, so 40 billion USD worth of drugs are falling off a cliff in the next 2 years. So this is a major, a big market size that is almost not explored sufficiently. What we can do, we can take any of these drugs and predict new indications and new combinations using our AI platform and create IP on the combinations and the new indications. What happens is that if we help and partner with those pharma companies, we can extend the patent life of those drugs 6-7 more years. And that enables pharma companies to keep the exclusivity of the market and keep the higher revenue. And not only that, we can also make their market size larger rather than let it stop by adding new indications, because these drugs are expected to work across many different cancers. And that partnership can generate revenue for us. The alternative is that we can run clinical trials and sell the therapy ourselves. Yeah, if you want to look at the cancer market compared to aging market, the cancer market is expected to be over 300 billion in 2030. However, if you want to look at how much it is worth to increase the lifespan of humans by 10 years, it is estimated to be over 300 trillion. These estimates are published in Nature journal, but that is more than an income of United States. Yeah, so we started off by our in vitro results. The aim was to find drugs that work across different cancers, at least starting with 2 cancers at a time. And we find 8. So these drugs had some features. Each of the combinations had to be better working than each individual drug and at lower doses, and that is what we call synergy. So these are gross inhibition of cancer. So we want to stop growth inhibition, stop growth of cancer up to 100%, but we couldn't do it by any individual drug alone. Also, the drugs that we chose are the ones that are going to go off patent. So, for example, Merck's drug is going off patent in 2028. We can help them to keep its high revenue and extend the market size. We can help any of these pharma companies mentioned, but that was our first run. We wanted something stronger that not only works on 2 cancers, but more cancers. So what we did is that we chose these 6 cancer types: lung cancer, pancreatic cancer, liver cancer, stomach cancer, kidney cancer, and leukemia. And we wanted something that works across 6 cancers. And each of these were representative of many more cancers in our clustering approach. And in our second run in vitro, we found 6 combination therapies that were active on all 6 cancers at the same time. And the most important thing that was that the doses that eradicated all cancers were below maximal safe tolerable dose. As I said, these drugs are all approved for subtypes of cancer, but not together. And because we know their safety profile, we know how much the concentration goes high in the blood and in plasma. So the doses we chose are below the maximal plasma concentration. And if you want to look at how much these drugs generate, the drugs generate 3 billion per year at the moment. By finding new indications, we can add seven more years to the patents. So which means we can save 21 billion USD for us and the patent holders. But that is only by saving the previous indications. But as I said, we are increasing market size, starting by 6 cancers. But the biggest aim after that is to design a cancer vaccine that is effective across cancers, and that is the biggest market size. We estimate that to be at least half of cancer market, reaching 150 billion USD. Yeah, so these are our initial in vitro results. As you can see, these doses are very, very low in nanomolar doses, below plasma concentration. And we could eradicate up to 100% of cancers, of lung cancer, pancreatic cancer, liver cancer, stomach cancer, kidney cancer, and leukemia cancer. And this is a combination of 4 drugs owned by these pharma companies. Each of these are approved for one subtype of cancer, but the combination is novel. But then the question was, is it safe? So the combination, we had to show that it is safe. So ex vivo, we tested that the safety level, but when I say ex vivo, we grow fibroblasts in 3D, and then we compared application of drugs compared to a positive and negative control. Positive control is something that is toxic. We know that is toxic on fibroblasts. You can think of it as a poison or acid. Negative control is just a vehicle. You can think of it as just water. And we had one way of measuring cell death that is a standard Dirac-7 measures how much cell death we have. So the drugs just slightly adjusted doses, very similar to that one, were showing toxicity just similar to negative control, just similar to applying water. And total HDF count is also similar and to negative control. And under microscope, if you look at the fibroblast cells, it's healthy and non-toxic at the concentrations tested. And this is case study 2, another set of 6, another set of drugs. This is again 4 drugs. Was killing up to 100% of cancer cells. Again, this was safe and non-toxic on fibroblasts. We repeated that 6 times, and this is the third combination, 3 drugs, and less effective on lung, but 100% on all others. And fibroblasts are healthy. Fourth example, and this is a 5th example, is only 2 drugs, and one of them is ConcR for lung cancer in another combination. But we found out that if you combine it with another drug that is only approved for one subtype of cancer, together it can kill 100% of cancer cells of all types. And that is just 2 drugs, 55 nanomolar or 50 nanomolar. And so if you look at those response curves at 10 nanomolar, we had like 20% of the cells remaining, and at the cancer cells, and at 50 nanomolar, almost all cancer cells died. And yeah, that is a sixth drug. It was slightly adjusted more, and we just reduced the dose of this one a bit more in the fibroblast tests. Okay, then what happened was that we tested the drugs ex vivo across 30 cancers. So that was the bigger aim. We wanted to show that the drugs work across 30 cancers. So what is ex vivo? Just to explain a bit more. So ex vivo is when you take patient-derived tumors and we grow it in the body of mice. And then we take that out, and in an environment where we have actual human immune cells, we want to grow it. And that is the best representation of what happens in the body. The tumor is 3D, is taken from actual patients, and we have human immunity. So that is ex vivo. I liked the way that CRO we are working with could really scale it to 30 cancers and produce results in a short period of time. So these are like 4 of our drugs, and on patient-derived tumors, the imaging after day 7. So the positive control, you can think of it as poison that it kills all cancer cells, and negative control is just like water. Just the cancer cells are grown. There is a big circle, and the cells are round and happy. But the positive control actually is where these are just remaining of dead cells. So the drugs after day 7, I want you to see that it is so much like the poison, even though we know they are safe on healthy cells, but they are like poison on cancer cells. And that was repeated for many cancers, 30 cancers. I'm just showing some of the ones that we could see with naked eye. This is the pancreatic cancer taken from a European person. So you see that all 4 drugs eradicate all cancer cells. This is gastric cancer, another deadly cancer. The 4 drugs are very effective, acting like poison. Uterus cancer, I think drug 4 and 1 are doing better. Still a little bit partial effect on drug 2 and drug 3. Drug 1 and 4 are doing very well. And this is colon cancer. You can see combo 4, combo 1, and I think combo 2 and 3 are doing pretty well, like the poison, like the positive control. We have lung cancer driven from a European person. Again, all 4 drugs are very effective. And melanoma, the drugs are again effective. This 1 and 4 are better, I think, than number 2. So as you can see, the drugs are working across cancers, and that takes us closer and closer to solving aging. That means that we are touching on key mechanisms that are shared across cancers. We will shortly have the full results on 30 cancers, and if you're interested, you can contact us for more details. What is our next step? So our next step is to develop a vaccine. So we have identified key targets, and we have identified, we have taken sequences of those key targets, and we also took sequences of all mutations that are very common in cancers. We have taken data of human microbiome and viral and pathogen databases. And all of that, we wanted to design a vaccine which has some features. Firstly, we wanted it to be safe to healthy human cells, not touching any human proteins directly. And we wanted it to create an immune effect that lasts for years. And we wanted it to be designed for all ethnicity groups, and we wanted to create immune response against all cancer types. And so the vaccine has all of these features. It is composed of some in-house algorithms that we developed, as well as some AI platforms for vaccine design. And the outcome of that is that we can create a peptide vaccine. And if you want to ask what is a peptide, it's a sequence of 15 amino acids that are sufficient for binding to the target of interest and creating the immune response. And that immune response is expected to eradicate all cancers of all types. And the vaccine, we expect to show it get approved for therapy as well as prevention for all cancers. And the goal is to live longer cancer-free. Well, if you want to look at our roadmap, we started with drugs with a broad effect across sterile cancers, and then we showed efficacy across cancers ex vivo. The next step is to design our vaccine and test we actually have designed our vaccine. We just want to test it ex vivo across sterile cancers. And what would we do next is to test it on humanized mice, the drugs as well as vaccines. The way we test on humanized mice for vaccines is that we immunize mice, and I should mention that humanized mice have human immunity in it. So we want to vaccinate humanized mice with the cancer vaccine, and then induce cancer and see that the cancer is not able to be formed at all. But the next step after that is to do IND enabling and run clinical trials. For vaccines, we would get approved for therapy first. But once it is out there, we can get it, try to get it approved for prevention as well. But after that, our mission is to scale this and create a vaccine for all chronic diseases, and just repeat these steps. Yeah, I want to just emphasize our advantages is, what do we do? First, we have combination of safe drugs, and this is an advantage. We have a validated AI platform that was validated in vitro in 11 cancers under a year, and we are targeting aging. And the consequence of having that is that because it is combination of safe therapies, we can have faster track to market. And because these are safe, we don't have to spend a lot on toxicity and higher probability of passing toxicity and phase
Maybe
1.
1 minute, Yasaman.
Yeah, and we will have a higher probability of success. And we have de-risked our business model because we run many drugs at the same time on many cancers, and we have higher probability of success for each. So we do it cheaper, faster, and target maximum number of patients. Professor Ranjan Nag, my co-founder, and he's a professional investor in 100 companies, and sold his companies to.
Okay, Yasaman, let's just close for questions.
Yeah.
Let's see. You can give the list. Yeah, you can give the list of the cancers. I guess one question is the peptide idea. If you
Yes.
don't have cancer, what will it bind to?
Yes. Yeah, basically, it tells your immune system and trains your immune system to see that this signature is cancer, and whenever you see it, you attack it. So if you don't have it, doesn't bind to anything. But it is training your immune system to, whenever any cancer cell wants to get formed in the first place, in the first cells, it just eradicates it.
What if so is this something you take once a year or just once, like smallpox?
Peptide vaccines are expected to give you immunity for years, 1 or 2 years. I think we would need a reminder after 2 years.
Okay, excellent. Let's now open up to questions from anybody. So you can use the raise hands function, or if you're shy, you can use the chat, or I will just pick up questions from the chat as well. So I guess Min's just asked a question about side effects. Are there side effects? Right. These are existing drugs, right? So they've got a safety profile, but maybe some of them still have some side effects. Can you comment? Yes, man.
Yeah, so the side effect of one of the drugs is reduced blood counts. That is very common in that is very common in anti-cancer drugs, but we are using lower doses, so we expect lower of that side effect.
Okay. These are questions for any of the speakers, by the way, or the speakers can ask each other questions.
So, Ranjan, I don't have a hands up function, so I'm just going to speak up if I may.
Yes.
So I really like the concept of this chronic disease, cancer being a chronic disease. We're starting to find out more and more about the microbiome and its importance. And if I can remind you of a discovery that was made in Australia about Helicobacter pylori and the inadvertent use of antibiotics as preventing peptic ulcer disease and gastric cancer. So why are we not working more on the microbiome and exploiting that?
Yes, man, you had microbiome on one of your slides. What's the referral there?
For vaccine design, when we design our vaccine, we just also look at its similarity of sequence to the whole microbiome, and that is included in our in-house derived vaccine design tool. So we do look at the whole microbiome sequences. Yeah.
Good question by Diana. I mean, I've got a COVID vaccine, and when I was a baby, I had this smallpox vaccine, this polio vaccine. And as you know, there is like an anti-vax movement out there. The idea is to give our immune system to something to practice on, but will we weaken our immune system by giving us all these vaccines? I guess that's the theory.
Yeah, is that a question?
That's a question, yes.
Yeah, so
comment on that.
yeah, I think we are working on peptide vaccine, and peptides length is very short, and we want to know that it is not interacting with any of human proteins. Today, it is easy to measure that by just running simple blast of the protein sequence versus all human proteins. And we choose the ones that are not directly interacting with any of human proteins. So peptide vaccines that we design are expected to be safe. But mRNA vaccines that are out there, so this is a very, very long sequence. And if you look at its overlap with human proteins, it's not 100% zero. So there was a question sometimes I was asked that a shingles vaccine increased the chances of getting dementia. And I looked at the sequence of shingles vaccine, and I looked, there is an overlap of that vaccine with nerve growth factor. And so that kind of made sense that the vaccine is interacting with some human proteins that we don't want it to Yeah, but I don't say we should definitely stop any vaccination, because some of the vaccines actually extend lifespan. So like 200 years ago, humans had a much lower lifespan, and what increased our lifespan from 30 years until about 76 years, which is now. Maybe one important contributor of increasing humans' lifespan might be advent of vaccines, and just a smaller amount contribution is due to drugs. So the vaccines help us to have a stronger immune system, but we should make sure there is no side effects to those vaccines.
So what's interesting in all three of these presentations, the general theme is to do it. I think Andrew said it basically, we run out of money basically. But all three of these presentations said, well, how can we get it cheaper? I guess like Yasaman's last slide, cheaper, faster, better. In trying. So Paul's idea was the AI, but try and make it pragmatic AI, right? Predict things, new things that are actually manufacturable. And then Andrew's idea was, well, can we actually get a higher incidence? Will it actually work for you? Which, we're all different. And then Yasaman's idea is sort of almost the opposite spin, is can we actually make it work for lots of other people? But how do we do it by not inventing a new drug? Let's just use the existing ones in different combinations. And I guess Sylvia's question is, and maybe Paul or Andrew can comment on this since Yasaman's been talking. Why aren't these drugs on the shelf? Why aren't they being dusted off? I think Paul had an example, one being dusted off. No, actually, I think it was Andrew, tamoxifen, which he said was found by accident, kind of thing.
Yeah, so
Why aren't we looking for more of that, more
yeah,
tamoxifen?
I tried to set up that business a while ago. It's just really hard to monetize and hard to police because I won't bore you with all the detail, but the road to market is very, very difficult. Even when they're close to the end of patent life, it's really, really hard. There's lots of drugs we use out there. We don't use in the right way. Lots of drugs that have a positive signal that aren't used. Lots of drugs that have been thrown off because there's no value associated with them. And it's very hard to do that. And despite governments saying this is what we want, they won't invest in it. So it's very difficult. And I've spent a lot of years of my life trying to do that, repurposing and reinventing. And unless the way we approach this licensing drugs changes, it's very hard.
I think I'd probably debate that, and part of this session is for us to have all the discussion. I'm probably voting with my feet and my dollars on the Agemica side. You could argue Ozempic is a repurpose. We've had that idea for a couple of decades, and now it's one of the most biggest revenue producing item in Norway. Obviously Denmark, I think. There is a science and legal art to patenting combinations. Now, of course, people can actually just start prescribing things off-label and themselves in combination once people realize. But there are other ideas. Let's see. Okay, we've got a few questions out here. Paul, yes, I think it's from Min, about a little bit more specialized on types of machine learning, GANs and adversarial autoencoders. I guess things popping up almost every day, I guess, Paul, on new types of ideas of machine learning. How do you keep up? And are you seeing something that maybe I'm not seeing that's particularly resonating?
Yeah, it's very difficult. I mean, there are indeed a great number of variations on a theme, and then one occasionally something that's a bit radically different. I guess like the adversarial networks was quite different from what came before, and now there are lots of different versions of that. I mean, my personal view, it's almost like language. I mean, you can get many of these things to work roughly equally well across many domains, but it's very specific. That I say with regard to the drug discovery domain, I think the issue is much less the fine details of the particular AI methodology, although some like the graph related methods I think do have some natural complementarity to chemical structures, which are you can represent as graphs themselves. So the mapping there is quite good. But I think that many of these things are just like other language. They could all get you to the right place if the input data is sufficient. So I think for in the chemistry domain, it's really the problems are around the input data, not whether a particular GAN or adversarial network will be better than some other architecture. I mean, those things do matter in the detail for maximizing the effect, but I don't think you'll see a great effect in one and no effect in the other, if that makes sense.
Okay, yeah, I guess if you look at the history of AI, people use tons and tons of computing power and complicated algorithms, and then eventually people start getting a bit cleverer and say, well, I can almost do the same thing without this complexity. That's probably not where we are yet. It's almost the other way around. But we will get more computing power every year. When do you think we can get Andrew's vision, or other people comment? He's just sort of AI at the desktop, AI at the bedside, where you're just doing instead of having the entire data center of Google at your disposal, you can actually do pragmatically in a regular rural hospital.
Well, Ron John, what's the comparator? The comparator is based on the clinician's decision, and it's a function of their intellect, their understanding, and their knowledge. And we've tested that, and the kappa comparison is like 0.15. You give the same clinician the same case 1 month apart, and they'll make a completely different decision. So the challenge we have is the comparator compared to what? Compared to what you get randomly.
So you're in the view you're in a teaching hospital, I guess. Is that right?
Yeah. But
so you're of the view that machines eventually will be better than doctors.
yes and no. So they will be better in being standardized, in interpreting complex data better, and they will not be mood dependent and emotion dependent. They will give you options.
Intoxicated.
Supporting data.
Okay, yes. Paul, go want to ask you a question about one way to prevent Yasaman's trying to prevent cancer before we get it, but another way is to detect it early, right?
Right. It's a different problem. It's a different set of prior probabilities and so forth. And so whether the same approaches for treatment of very early stage cancer is the same thing as preventing the risk of cancer. I don't know the answer to that. I'm interested. I suspect there's AI approaches to both problems, but they are both relevant. And the surgeons like to think that they treat early stage cancer better than all these drugs anyway. So take it out, and you're cured. Now, naturally, your risk for next cancer is higher, but in many cases, but it's still. Those are still separate problems.
So, Andrew, you're a surgeon, or you teach surgery.
Yeah.
What's your view on that?
Paul, we could talk for hours about this, but we haven't made a lot of traction in the early detection of cancer. Where we make the most traction is the early detection of precursor lesions of cancer, cervical cancer screening, colorectal cancer screening. If you look at even in the early breast cancer data, it's not because they're treating invasive cancer that they've detected DCIS.
Right.
So it's the early detection and treatment of precursor lesions that makes all the difference.
Pre-cancer. These are what we call precancerous
Precancers, yeah, like the cervical screening was intraepithelial neoplasia.
So do you recommend, usually radiologists don't recommend it, but I'm quite active on it, a full body MRI scan every
Yeah,
1
well,
year or
Ron,
two?
John, go ahead and I'll operate you, charge you like anything for taking bits and pieces out of you, and it probably won't do you any benefit apart from, like my new Ferrari.
And most of what he'll remove is normal.
Well, no, I wouldn't
Exactly.
remove it. What I might do, I think my understanding is you take the snapshot, you do it a year later, and if it's not moved, then don't worry about it.
Yeah,
Right.
well, Ron, John, I can halve your risk of kidney cancer, testicular cancer, lung cancer, uh, any other cancer that has more than one organ. So if we take your kidney out and we take one testicle out, we take this out, you get a halve your risk of cancer.
And you end up
Yes.
bionic as well, so that's all good.
Well, if you take your testicles out, then your thymus starts to regrow. So, which beefs up your immune system. But most people don't want to do that. This is a great question from Dow, maybe for Yasaman. What about, can we build a vaccine for an AI for a disease we actually don't know about yet?
Yes. Yeah, the good thing about my approach is that we don't need huge amount of data.
No. What if you just don't know? Like COVID just pops along. We didn't even know
Yes.
that it existed.
We
That,
just need gene expression profile of like patients getting COVID.
oh no, but what if you don't? This is before you get COVID. This is before the virus actually exists.
So basically, for AI, you need to have something. We don't need vast amount of data, but we need a little data.
Now, that's my next challenge for you. My next
But yeah,
challenge.
it has to make decisions based on something. It makes decisions based on genes and pathways that are disrupted.
So my next challenge for you and the team and Paul and Andrew is yes, drugs and vaccines for diseases we actually don't know about yet. So I can imagine philosophically a screen for things that are good and everything else that's not good.
Yeah, for vaccines.
Moonshot idea. No,
Yeah,
with no
vaccine.
data training. So Andrew's got quite far ahead. He's trying to do it with little data. I want you to come up with a solution where there's no data.
Well, vaccines, as I said, vaccines, when you have a vaccine with such a profound effect across chronic diseases, I suppose it would protect against other diseases we don't know about, because as I said, we are targeting key mechanisms of aging. So any other disease of aging that we don't know much about, once we develop the vaccine that is for aging, it prevents other diseases of aging that we even didn't think about in the first step.
So that was the point about it, because somebody talked about comorbidities, and that's what you're kind of seeing with some of these. It's like cause and effect with some of these latest weight loss drugs, and people are saying, well, actually, it fixes cardiovascular as well, and it fixes reduces the chance of cancer and reduces all these other diseases. And my argument is not because of the actual Ozempic drug or the GLP-1 inhibitor, it's because of the weight loss. So yet to be tested, but it's not. There are other ways to have weight loss.
Yeah.
To
If
reduce
you have
disease.
Vaccine for diabetes, blood pressure, and obesity, and all other chronic diseases you can think of, and remove all the risk factors, then we are reducing chances of getting any chronic disease as well.
All right, let's see. We've got 2 minutes. Somebody who wants to ask a question, one more question, if they want to ask. Otherwise, I'll make the statement. So all these. Okay, go ahead. Yes.
Josephine.
Yes, I want to ask this. The last presentation was about aging, which is very important, and the common consensus was that this was a disease of aging. But we're seeing more and more young adults being diagnosed with various types. This seems to be sort of like the new arena, and what's the focus on that? And what's triggering cancer earlier and earlier, and various types?
Who
Yeah,
wants
that's a really interesting question because there's a number of factors. People have postulated a lot of things, including what's called the cohort effect. Every next generation has an increase in the risk of cancer, and we don't understand that. Whether it's more exposure to chemical, whether it's differences in diet, we see clear steps in the way things have changed over the years when we move from pre-processed wheat and other things to processed things. The incidence of colorectal cancer went up. We can see some of these increments, but we can't really answer that because of the cohort effect, that every new generation seems to have a greater risk of cancer. And it's a known phenomenon that younger and younger people are getting cancer, and there's data to support that that's the case epidemiologically, but we don't really know the cause of that.
Excellent response, Andrew. Maybe the vaccine idea will help. Maybe Paul will invent a drug for us as well. If you want to contact anybody here, you can either do it through me or each of you can type your email address in the chat as well. And we will send a survey out. All these companies have investment opportunities as well, if anyone's interested in that. And if you're interested in various ideas and the various products, we'll have more of these sessions as well. Many of you are familiar, and hope to see you in subsequent events. Thank you very much. Thank you, Andrew.
Thanks,
Thank
Ron
you,
John. Thanks
Paul.
for the opportunity.
Thank you.
Thank you,
you.
Yasaman, and it'll be recorded. It'll be on YouTube as well.